All right, hello, and welcome to the reproductive system and development. This video is going to cover the male reproductive system, and then I will do a second video for the female reproductive system. So the male reproductive system is made out of many parts, some of which we consider the primary sex organs, some are accessory sex organs, and primary sex organs are the gonads, what makes the actual gametes, and that would be the testes in the male. So we have the testes here in the scrotum. The scrotum is the sac that hangs outside of the body. It has smooth muscle in it. The smooth muscle can contract and relax in order to help regulate the temperature of the testes. The testes do need to maintain a lower temperature than normal body temperature in order for sperm to develop and mature. So therefore they are on the outside of the body. From the testes, we can see that there is the epididymis that sits on top of the testes. There's the ductus deferens that comes up and around the urinary bladder and then connects with the urethra, which is the exit out the end of the penis. So on the penis, about half of the penis hangs outside of the body and the other half is internal. There's some tissue in there that we'll get to later and discuss. The end of the penis is called the glans of the penis, and then the prepuce is what is on the outside of the glans of the penis, and sometimes it's known as the foreskin. So go ahead and pause the notes as you need to take down the information that I just covered. This here shows the seminiferous tubules. The seminiferous tubules are small tubes that are in the testes, and this is where the sperm are made from stem cells. Those stem cells start out and undergo a process that takes about 10 weeks for the sperm to develop. Those tubules are filled with fluid that are going to help move the sperm as the developmental process continues, and the sperm will continue to develop in the epididymis before they end up mature and ready to fertilize an egg. In between the seminiferous tubules, which these circles here are the seminiferous tubules, you're going to find Leydig cells, or what we would call interstitial cells. This is where we're going to have the secretion of testosterone come from. So testosterone, of course, is the male hormone, and that is going to come from those cells that surround the tubules. Okay, so here you can see the sperm in more detail. Of course, the diagrams always show us more detail. And we know that the sperm is going to develop over time from stem cells. So I'm going to go through and show you the process here of how that happens after you record the notes over the testes. And go ahead and pause this if you need to write these down. Okay, so here is a sperm. A sperm is made up of three main parts: the head, the midpiece, and the flagellum, or the tail. The head is special because it has an acrosomal cap, which has digestive enzymes, which will help the sperm penetrate the egg. Inside the nucleus, you're going to find those 23 chromosomes. The mitochondrial spiral is going to project and cause the locomotion, which will be the movement of the flagellum or tail. So when we're looking at the development of sperm cells, it is difficult to see, but we want to look at the different layers here because we have stem cells around the outermost layer of the seminiferous tubule, and as the cells develop, they get pushed more towards the lumen, and the lumen would be the space in the tubule. So these little dark spots that we see right here, these are the sperm developing. And you can't really see them in this picture here, but we definitely can see them here in this design where the tails are sticking out there, okay? And what we're seeing here is we're seeing these cells here at the top. Those are going to be our spermatogonia, or the stem cells, and we can see here that they have a full set of chromosomes still, 46, okay? And when they reproduce, we end up with primary spermatocytes. And those primary spermatocytes, as you can see moving down, they still have 46 chromosomes, okay? And then they turn into secondary spermatocytes when the number of chromosomes is halved to 23, okay? So as they move down, they become early spermatotids and then late spermatotids, and they grow their tails. And once they are past that late spermatotid stage, they then will break away and be flushed through the lumen of the seminiferous tubule to the epididymis. So again, secondary spermatocytes are going to have 23 chromosomes, or N. Okay, the primary spermatocytes had that full 46. So that right there is where meiosis 1 is completed. So in your lab, you're going to be looking for the spermatogonia. You're going to be seeing those primary spermatocytes. We're then going to see where those secondary spermatocytes show up, where the spermatids are, and where the sperm cells are about to detach from the inside of the seminiferous tubules. I do want this slide here in your notes, so go ahead and pause and take those notes down. So now the sperm, Sertoli cells. Make sure you write down the Sertoli cells. They protect and nourish the sperm cells throughout the developmental process. We are going to be looking for Sertoli cells in our lab, so. Okay. So the sperm are carried from the seminiferous tubules down and into the ducts of the epididymis. And it's here that it takes about 20 days for the sperm to develop. This is when the sperm learn to swim, and they are then ready to fertilize an egg. So during sexual arousal, the sperm will move, and they will be ready to be ejaculated. They will travel up the ductus deferens. When it comes past here, the seminal vesicle is going to release fluids, which will add to the sperm. The prostate gland is another part here that is going to add fluids to the sperm in order to make them able to swim, okay? And then the bulbourethral gland is also going to add fluids that will help the sperm in order to make their way towards the egg. All of that fluid will be ejaculated out of the end of the penis, and okay, so we are going to now continue on to the parts of the penis itself. There are 3 columns of erectile tissue, and those erectile tissues cannot be seen from the outside here. So we have to travel inside, which here is the penis here. We have the purple columns right here, which are represented down here by the corpora cavernosa. And then we also have the corpus spongiosum. And these are going to be the spaces that are going to engorge with blood during an erection. So again, the glands of the penis can be covered with the prepuce or the foreskin, and circumcision is the removal of that flesh, that tissue. Okay, next we are going to talk about the glands. the glands and the seminal vesicles, the prostate gland, and the bulbourethral gland. So these three glands add nutrients to the semen. They change the pH so that way the sperm can live, and they lubricate the urethra and neutralize the pH in the vagina so the sperm have a chance to live. So next, the secretions that are made. The secretions that are made, we call them semen. The semen is going to be a mixture of that sperm and secretions from the glands, and we call that fluid as it leaves the body an ejaculation. It's a forceful expulsion of semen. It comes out the urethra. It's propelled by smooth muscles and skeletal muscles. Now that ejaculatory fluid is very special because there is between 2 and 5 milliliters of ejaculatory fluid, and in that ejaculatory fluid there are about 250 million sperm. If a man has less than 20 million sperm per milliliter, he would be considered not fertile. Sperm is only 1% of the ejaculatory fluid that is released. There's fructose, amino acids, vitamins, calcium, proteins, sodium, zinc, and citric acid also in ejaculate. So this concludes our portion of the male reproductive system. Make sure to end this section with a summary of the male reproductive system, and there is a second video on the female reproductive system.